Right now we're studying 12.3 DNA replication. DNA is copied, as you can see, as we've talked about over here in the S phase, synthesis. What's being synthesized in the S phase is DNA, a second copy of the DNA, so that when the cell splits over here during mitosis, there's another set of DNA. So both cells have a copy of the genetic material. It's contained in the nucleus. That's the job of the nucleus, is to protect the DNA. And because DNA stays in the nucleus, that's where it has to be replicated. That should make a lot of sense. So because DNA stays in the safe nucleus, replication occurs right there. The process is called replication because we're making a replica, or an exact copy of the DNA. The part of the DNA that allows for this is the weak hydrogen bonds. If you remember the structure of DNA, there is, if these are the two parallel strands, anti-parallel strands, there's a weak hydrogen bond which allows for the molecule to be split apart, just as we see right here. So enzyme helicase is the enzyme that does the job of splitting open DNA at the weak hydrogen bonds. So remember that enzymes are proteins that have a job, and they tend to end with the letters ACE, A-S-E. So enzyme helicase unzips the DNA, so it's represented by this purple triangle right here. And where the DNA is being opened is called the replication fork. So you can see it sort of looks like a fork. Here's the top, the part that you hold, and this is the part where it goes down and it stabs into your food. So that's the replication fork. The original strand that is being copied is the template strand. So if this is our original strand right here, so this is the original band of DNA, and then it's unzipped by enzyme helicase, which we see here. The original half is here and here, and then nucleotides are being added in the middle. So these are the complementary strands. So at the end of DNA replication, you have two strands of DNA. Both are going to be half original and half new, half template and half complementary. DNA polymerase is represented by this little yellow square here. It joins free-floating nucleotides that are around the replication process. It takes those and matches them up with the complement of each of the nucleotides. So if on the DNA it says A, it'll find a T, and if there's a G, it'll find a C, and it attaches it in this 5' to 3' direction. Replication in prokaryotes there's one replication fork, and then the enzymes move in opposite directions until it finishes the plasmid, or the circular ring of DNA. And this doesn't happen in the nucleus because prokaryotes don't have a nucleus. They do have a nucleoid region, though. In eukaryotes, there are many replication forks. We're a more complex cell, and so therefore more enzymes to do more jobs and to get this job done faster. Eukaryotes have lots of DNA for its complexity, so it pounds this out really quick with multiple replication forks. So what we're going to do is we're going to practice some of the complements. So if we have a strand of DNA and the strand says GATTC, DNA polymerase is going to match a C, a T, a T, an A, and a G. So remember that A always matches with T and C always matches with G. Let's go to the next page. Alright. Here we have C, 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 T, G, A, G, T, A. So this is the template strand. This is what the original DNA strand says. And the complementary strand that DNA polymerase is making is going to be G. It finds the free-floating nucleotides and assembles them. G, 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 A, C, T, C, A, T. And one last one. Here we go. So C becomes G, G, G becomes C, G becomes C, T becomes A becomes T, C becomes G, G becomes C, A becomes T, T becomes A, T becomes A becomes T. And that's it. That should get you through until we talk about RNA synthesis. Have a great day.